I'm going to be talking about the long-term effects of cannabis on cognitive function, but I'm going to extend this also to studies of the long-term effects of cannabis on the brain, on brain structure and on brain function, and also placing this into the context of cannabis and psychosis, given the increasing interest in an association between cannabis and schizophrenia. And you might see there my rather intriguing subtitle of The Good, the Bad and the Unknown, and hopefully that will become a little clearer to you as the talk progresses. You'll have to excuse me as well, I have a bit of a cold, so if I cough or sniffle, please excuse. Okay, so starting first of all with the context of cannabis and psychosis. My slides are just not progressing, here we go. Okay, so many longitudinal and epidemiological studies have now shown that cannabis use increases the risk of developing psychotic symptoms. The evidence suggests that cannabis use may in fact double the risk of developing schizophrenia, and some say that that is in fact a conservative estimate. But essentially, the greater the extent of cannabis use, the greater the risk, and particularly when it's high-potency cannabis that's being used on a daily basis over the long term. So, can we say that cannabis causes schizophrenia? Well, the answer to this clearly has to be no. There are millions of cannabis users worldwide, and only a very small proportion of them actually develop psychosis. Now, we don't know what are the causes of schizophrenia per se, and we clearly need a lot more research to understand how cannabis interacts with a vulnerability to psychosis. What we do think is that cannabis is a component cause of schizophrenia. That is, that it combines with a range of other risk factors to trigger the disorder in vulnerable individuals. And what we really need now is research to understand what it is that makes an individual vulnerable to developing psychosis if they use cannabis, and the mechanisms by which cannabis might cause psychosis. So these are the research questions that have been driving our program of research in recent years, but the entire body of research that we've done over many years looking at the effects of cannabis on cognition and on brain function, we hope will also provide the background towards greater understanding of this question. So most of the studies that I'll be talking about today are studies of chronic cannabis users, where we've used a range of methods, including psychophysiological, recordings of brain electrical activity, neuropsychological and neuroimaging techniques to assess cognition and brain structure and function in long-term or heavy cannabis users, sometimes in short-term and light cannabis users, and in all cases we match our cannabis users with non-user controls on a range of critical factors such as age, sex, IQ, education, as much as possible alcohol and other substance use, sometimes personality measures, such that the main thing that differs between the groups that we want to compare are their level of cannabis use. And we mostly test people in the unintoxicated state. That is, mostly they are long-term or regular cannabis users, but they're not stoned at the time of testing. And we think that the state in which we are testing people represents the state that chronic cannabis users are operating in on a daily basis when they're not intoxicated. More recently, we've been conducting some studies of acute administration of cannabis to humans, and I'll finish my talk by describing some of those studies. So to cut to the chase, the research from our team and many other labs around the world now does indeed suggest that long-term or heavy cannabis use results in cognitive dysfunction that persists beyond the period of acute intoxication. This cognitive impairment can last for hours, days, or months, and has been shown to be related to frequency, quantity, and duration of cannabis use, and importantly and increasingly in many studies now, has been shown to be associated with the age of onset of cannabis use, which really points to the sensitivity of the adolescent brain to the adverse effects of cannabis. And I'll talk about this at several points within my slide. So if cognitive impairment exists, does it recover after abstinence? Well, the answer to this is really uncertain. There's one study that everybody loves to cite that was conducted by Pope and colleagues in 2001 that showed recovery of memory function in particular after 28 days of abstinence. But there are many, many other studies that show persistent effects of cannabis on cognition and on more subtle measures of brain function beyond 28 days of abstinence. And really, the definitive before and after study has not been done. Now, many of you may have heard about this study by Meyer and colleagues on the Dunedin cohort following the sample longitudinally over many years that showed a persistent decline in IQ amongst those who had stopped using cannabis or greatly reduced their use, but had started using during adolescence. So again, the sensitivity of the adolescent brain to the impact of cannabis. Now, we really think that IQ is more of a global measure, but if we look at specific cognitive domains, we believe that some of these should in fact be amenable to recovery. And that's based on what we know about the plasticity of the human brain. But again, I emphasize that the definitive studies have not been done. We really need to do a lot more research into recovery. I have a few more things that I'll say about that later in the talk. So if cognitive deficits exist in cannabis users, what is the nature of this cognitive dysfunction? Well, mostly we see impairments in the domains of memory, attention, executive, and higher cognitive functions. And over the years, we've started to notice a similarity between the deficits observed in cannabis users and those observed in schizophrenia, albeit to a lesser degree in the cannabis users. So when we examined the literature, looking at evidence that links cannabinoid function and effects to schizophrenia endophenotypes, let me just focus on what are the endophenotypes. Endophenotypes are internal markers that are thought to be intermediate between the genotype on the one hand and the phenotype, the very complex presentation of schizophrenia with heterogeneous symptoms, etc., on the other hand. It's thought that by focusing on endophenotypes, we might get closer to understanding the causes, the risks, the vulnerability to the development of psychosis. So endophenotypic markers, such as these cognitive domains, have been shown to be impaired in first-degree relatives of patients with schizophrenia, that is, unaffected relatives, and it's thought by focusing on these domains, we'll understand more, as I said, about risk markers for schizophrenia. So when we looked at the literature in cannabis users and in animal studies of administration of cannabis, etc., we found evidence for impairment in the functioning in cannabis users across most of these endophenotypic domains. We found evidence for the direct involvement of the endogenous cannabinoid system in the brain from animal studies, and we found evidence for neural substrates within the brain that interact with the endogenous cannabinoid system across all of these cognitive endophenotypic domains that have been identified in schizophrenia. Now, in some ways, this is really not that surprising if we consider the distribution of cannabinoid receptors in the human brain, where we see high density of cannabinoid receptors throughout regions of cerebral cortex, in the cerebellum, in the hippocampus, in medial temporal lobe, basically in also prefrontal cortex, areas that are associated with higher cognitive functions. Now, the endogenous cannabinoid system in the brain is essentially a retrograde messenger system that's involved in regulating neurotransmitter release, and particularly in regions that are involved in cognition and also implicated in schizophrenia. The endogenous cannabinoid system is largely an inhibitory system, so it inhibits the release of these major neurotransmitters. We know that with acute administration of cannabis, there is an increase in frontal and striatal dopamine metabolism and release. Now, striatal dopamine metabolism and release is particularly interesting because it could perhaps explain the way in which cannabis might trigger psychotic-like symptoms. But we also know that chronic administration of cannabis leads to a persistent and selective reduction in prefrontal cortical dopamine turnover, and this could explain some of the cognitive deficits in cannabis users. Now, over the years, increasingly, we're seeing many more studies being published in journals such as Nature Neuroscience, etc., that increasingly look at the specifics of the role of the endogenous cannabinoid system in the brain, particularly on hippocampal function involved in memory, and looking at ways in which exogenous cannabinoids, that is by smoking cannabis, how that impacts and perturbs the endogenous cannabinoid system to affect some of these, particularly memory processes. So we know that endogenous cannabinoids are critically involved in the induction of LTP, long-term potentiation, which is the cellular basis of learning and memory. And we know that they interact with NMDA receptors and the glutamatergic system. So research has shown that chronic exposure to cannabis disrupts long-term potentiation and NMDA receptor function. And our most recent studies looking at acute administration of cannabinoids to humans are increasingly focusing now on this system. And aside from the dopaminergic hypothesis of schizophrenia, which has prevailed for a long time, there's now increasing evidence in a glutamatergic hypothesis for schizophrenia. So the fine-tuning role of the endogenous cannabinoid system can be deregulated by this potent and less selective bombardment by THC acutely. And with acute intoxication, we see cognitive impairment, mild hallucinations, delusions, and perceptual distortions. We think that in the long term, with chronic exposure, this may result in lasting dysfunction of the endogenous cannabinoid system, schizophrenia-like neurotransmitter conditions, desynchronized neural networks, psychotic symptomatology, and cognitive impairment, again, primarily in the domains of attention, learning, memory, and executive functions. Now, I'm going to turn to verbal memory for a moment because this is one of the most impaired cognitive domains in schizophrenia, and it is the most consistent deficit associated with both acute and chronic cannabis use. So there have been multiple studies of long-term or heavy cannabis users that have shown impaired performance on word list learning tasks, such as the Ray Auditory Verbal Learning Test and other variants, and functional impairment has been demonstrated in neuroimaging studies of verbal learning in cannabis users. So the evidence cumulatively suggests largely impaired encoding, but also impaired storage, manipulation, and retrieval mechanisms in long-term or heavy cannabis users. So this slide shows you the typical pattern of performance on one of these word list learning tasks. So here we're looking at long-term versus short-term cannabis users versus controls, and other studies have shown very similar patterns when looking at heavy versus light cannabis users and controls, where the long-term or heavy cannabis users learn fewer words across the learning trial, they recall fewer words, and they forget more words after interference or after a delay. So we have observed this to be more associated with the duration of cannabis use, and so have some other groups, whereas other studies have found this to be more associated with the frequency of cannabis use, or perhaps with the dose, the number of joints per week. And it's precisely on this task that the Pope study in 2001 showed recovery after 28 days. Another study has shown no recovery after 28 days abstinence, and we've seen evidence of some partial recovery with cessation or reduction of cannabis use. But what was interesting was when they re-examined their data, they found that recovery was actually less apparent when the age of onset of cannabis was prior to 17 years. And there have been a number of other studies now also demonstrating greater adverse effects amongst those who commenced using cannabis prior to age 16 or 17. Now, we and one other group have administered this task to a sample of adolescent cannabis users, and we found that performance in the adolescents worsened with quantity, frequency, duration, and age of onset of cannabis use. So the younger one of the things that was particularly striking was that this sample of adolescent cannabis users, they'd only been using cannabis for about 2 to 3 years, and they had only used cannabis about 2 to 3 times a week. So not a really very heavily entrenched group. And nevertheless, the degree of impairment that we observed on this task relative to the adolescent age-matched counterparts, non-users, was similar to what we saw in adult cannabis users who had been using pretty much daily for about 20 years.
Excuse me, Nadia?
Yeah.
Apologies for the interruption. We seem to be losing your phone. It seems to be coming in and out.
Oh. I'll try going off speaker and onto handset.
Okay.
So let me know if this is better.
Yes, that's much better. Thank you.
Okay. Yes, we observed a similar degree of impairment between the adolescent cannabis users, despite them only using 2 to 3 times a week for about 2 to 3 years, and they were about age 18, to what we saw in adult cannabis users who'd been using for about 20 years on a near daily basis, but didn't observe in adult cannabis users who had used for up to 10 years. So this suggests indeed a greater sensitivity of the adolescent brain to the adverse effects of cannabis. Now, what we think is going on there is adolescence is a really critical period of neurodevelopment that involves significant resculpting of the brain from the ages of around 12, 13, 14, continuing through teenage years, up to 18, 20, even up to 25, with still the development of prefrontal cortex in the early 20s. So we think that cannabis use coming in to bombard the brain at these critical periods of development might be what explains greater adverse effects. And of course, that particular period of development coincides with the time of onset of schizophrenia, where things are going wrong with the development of the brain, with apoptosis, with pruning, with white matter development, with pruning back of grey matter. So it's a very critical period to focus on in terms of understanding long-term effects of cannabis. Now, I might say that this doesn't really apply across the board. So in some as yet unpublished data, we administered the Cambridge Neuropsychological Test Automated Battery, the CANTAB, to adult cannabis users and adolescents. Now, in adults, we saw impairment pretty much across the board. Mostly these were tasks of memory or working memory, vision tasks. In adolescent users, we only saw selective impairment. So performance was intact on a number of tasks, but impaired on others. So what we want to do now is to really look at these data more closely in terms of these neurodevelopmental trajectories in the brain, at what point cannabis use bombards and may cause greater adverse effects than what we see in adults, and of course how that develops over the lifespan with continued cannabis use. Okay, so since then we've gone on to try to understand what is going on in the brain of cannabis users when they're performing a range of cognitive tasks. I'm not going to go into detail on this slide, but we have used electrophysiological markers, brain event-related potentials to look at selective attention, early sensory memory, sensory gating, and verbal learning. So we can see patterns of electrical activity when people correctly recall words compared to when they forget words, etc. We've seen associations with the duration of cannabis use, with the age of onset of cannabis use, and we've also seen some of these very sensitive markers persisting in samples of ex-cannabis users, which suggests perhaps lack of recovery or only partial recovery. We've moved on from those kinds of studies to some brain imaging studies, again mostly focused on verbal learning and memory. And we've seen regions of medial temporal lobe, dorsolateral prefrontal cortex, insula, hippocampus, that are differentially activated between non-users and heavy cannabis users. And we're now considering the brain more in terms of it being a network. So rather than just focusing on specific brain regions, we're looking at connectivity between regions in the brain. And in a preliminary analysis, we've seen aberrant connectivity between the hippocampus and different regions of the cortex. And in fact, what we observed in the cannabis users was increased connectivity, suggesting that cannabis users have to work harder to try to recruit the brain regions responsible for performing these kinds of tasks. So again, functional connectivity, with functional connectivity, we can look at cognitive task-related changes in the strength of interaction between two or more brain regions. And this is again looking at functional connectivity, this time in a cognitive control task that involved response inhibition, avoiding distracting stimuli and incongruent stimuli. And again, we saw increased connectivity between different brain regions, suggesting that cannabis users have to really work harder to recruit these brain regions in order to perform the task. Now, aside from functional connectivity, we can also look at structural connectivity. And we've done so by using diffusion tensor imaging and tractography to trace the white matter axonal fiber tracts throughout the brain. So this technology generates millions of different connections throughout the brain. And in a whole brain analysis, we found only 2 but very specific fiber bundles that were significantly reduced between 84 and 88% in long-term heavy cannabis users. So what this suggests is that connectivity and communication throughout the entire brain is not disrupted in cannabis users. It is very specific, and again, it pointed to disturbances in the hippocampus. So we saw an 84% reduction in the fiber bundles connecting the right fimbria of the hippocampus through the hippocampal commissure and the splenium of the corpus callosum bilaterally. And we saw an 88% reduction in commissural fibers connecting the splenium with the right precuneus. Now, these are very dramatic reductions. And this was in a sample of very long-term, very heavy cannabis users, again, about 20 years on a near daily basis. And the cannabis users were impaired in terms of their memory function, and they did display elevated depressive, anxiety, and psychotic-like symptoms. And yet we didn't see any direct correlation either between the memory impairment or those symptomatic measures and these specific reductions. In some ways, that's not entirely surprising because those kinds of functions, memory, performance, and symptoms, arise from a range of communications throughout the brain, and it might be a little surprising for them to be solely associated with these very specific regions. Nevertheless, it's an important finding that I think we really need to follow up on to fully understand the functional and clinical significance of these findings. Now, aside from connectivity in the brain, of course, we can also look at volumetric measures of specific brain regions. And one of our major findings several years ago now was in dose-related reduction in hippocampal and amygdala volumes in long-term heavy cannabis users. So we found 12% reduction in hippocampal volume bilaterally in the cannabis users compared to non-users, and we also found a 7% reduction in amygdala volumes bilaterally. Now, just to put that in context, what is typically observed in schizophrenia is about an 8% reduction of hippocampal volume. So schizophrenia is characterized indeed by reduced hippocampal volumes, but only by about 8% generally. We found a 12% reduction in the cannabis users. So the cannabis users were indeed very heavy, 20 years on a daily basis, approximately 7 joints per day. And we found that the left hippocampal volume reduction correlated with the cumulative dose of exposure to cannabis. So you can see that here in this scatter plot. But what's more so is that we also found that it was associated with the development of subclinical positive psychotic symptoms. And the subclinical positive psychotic symptoms also correlated with the cumulative dose of exposure to cannabis. So this raised a number of issues. Now, our sample were very carefully screened for psychotic disorders, and yet they developed memory deficits, brain changes, and subclinical positive symptoms similar to what is observed in schizophrenia. Now, they also developed some significant subthreshold negative psychotic symptoms and elevated depressive symptoms, but we didn't find that those were associated with hippocampal volumetric reductions, only the positive psychotic-like symptoms. Now, the mean age of our sample, around age 40, suggests that they were not in a prodromal state, that is, the state before the development of an overt psychosis. And this suggests that, in fact, and made us realize that not only in this study, but in many studies, what we had been studying was those that are resilient. So these people, in many of our studies, have been very long-term and very heavy cannabis users. And if they hadn't developed schizophrenia earlier in their drug-using career, it's likely that they actually never would. So they have developed memory deficits, brain changes, and symptoms that are similar to what's seen in the brain of people with schizophrenia, but they haven't developed an overt psychosis. So that means we can start to ask questions about resilience. There are two questions. We can ask what makes a person vulnerable to developing schizophrenia if they use cannabis, what makes a person vulnerable to developing these kinds of changes which are a schizophrenia-like brain and yet not develop schizophrenia, and so what are the protective factors and what constitutes a resilience to developing psychosis? So why didn't they develop psychosis earlier in their cannabis-using career? Well, we scoured our data fairly closely looking for clues, and the only thing that really jumped out at us was the age at which they started using cannabis regularly, which was around age 20. And in the scheme of things, that's quite old. So we wondered whether that in itself might be a protective factor, the fact that this sample in particular didn't start using during adolescence. But we did have a few that started using as young as age 12 or 13. So then we turned to perhaps genetics as a possible explanation. So this important paper by Caspi and colleagues was published in 2005 and created quite a splash in literature. They looked at the COMT gene, which is associated with dopamine metabolism, and found that those with a particular variant in the COMT gene, which is actually very common in the general population. So in the general population, approximately 1/3 of us have each of these types of variants in the COMT gene. And they found that those with this particular VAL variant were significantly more likely to develop a schizophreniform disorder during adulthood if they had used cannabis during adolescence, but not if they started using cannabis during adulthood. So this was quite exciting and led to a number of other studies which unfortunately did not replicate these findings and started to implicate a range of other genes, including the AKT1 gene. But the COMT story is not dead. So over the last few years, we've seen multiple studies coming out further implicating the COMT gene with an association with psychosis when cannabis is used, with greater psychotic-like symptoms, and also greater cognitive impairment in cannabis users. So we've been starting to build up a database of cannabis users for whom we have DNA samples and controls for whom we have DNA samples, and we have this range of different measures of brain structure, brain function, electrophysiology, memory function, and symptomatic measures of psychotic symptoms, depression, and anxiety, almost all of which differ between our cannabis users and controls quite significantly. And we're starting to try to pull out genes that might explain why people might become more vulnerable to developing deficits in these range of phenotypes, and also what might indeed be the protective factors. Now, I might just say that we have since replicated our findings of hippocampal volume reduction in cannabis users in a much larger sample, about 70 cannabis users, and again found a reduction of hippocampal volume, this time by about 11% in right hippocampus, but only 6% in left hippocampus. And amygdala was again reduced by about 7% bilaterally. So these very heavy cannabis users, again, showed memory deficits. They showed elevated symptoms, anxiety, depressive, and psychotic-like symptoms, poorer global functioning, that in this sample we did not find a direct association between any of these outcomes and reduced hippocampal volumes. We did, however, find an association between reduced hippocampal volume and age of onset of cannabis use, and also some markers of neurochemical markers in the brain that we can measure through magnetic resonance spectroscopy. So we started looking at some of our genotypes in association with those outcomes, and we did find an association with the COMT gene for hippocampal volume and also for one of our electrophysiological markers, the mismatch negativity. But no, just as schizophrenia will never be explained by a single gene, similarly, no single gene will explain greater adverse effects of cannabis, and genes interact with multiple other genes. So when we start to look at epistasis, that is gene-by-gene interactions, we see significant interactions between the COMT gene and a range of other genes, also the AKT1 gene and a range of other genes. Now, the trouble with these genetic type studies is one really does need to have very large sample sizes, and perhaps the 200-odd people that we currently have DNA on is insufficient, but we're continuing with this stream of research in future studies. Okay, now we've also looked at some other brain regions in cannabis users, and now also incorporating patients with schizophrenia who have and have not used cannabis. When we looked at the cerebellum in particular, white matter in the cerebellum, we found a 30% reduction in patients with schizophrenia who also used cannabis, a 24% reduction in otherwise healthy cannabis users without schizophrenia, and an 18% reduction in patients with schizophrenia who didn't use cannabis relative to controls. So this suggests a kind of a hierarchy with additive effects of cannabis use and the pathology of schizophrenia, but also suggests that perhaps it's cannabis that's causing the greater alteration to cerebellar white matter than schizophrenia per se, given these differences that we've observed. And we've seen a similar hierarchy of effects when we looked at hippocampal shape, another way at looking at how the hippocampus is altered. And again, we found that the schizophrenia patients with cannabis use showed the greatest alteration in hippocampal shape, followed by healthy cannabis users, and then the schizophrenia sample without cannabis use. And this accords with other studies in the literature that have shown greater total brain grey matter loss over 5 years in a first episode sample with psychosis who used cannabis. So that was, the data I've just shown you focuses on brain structure, but if we look at cognition, we see a somewhat different story. So over the years recently, we've been seeing a number of studies coming out suggesting that patients who use cannabis show better cognitive performance than patients who don't. Most of those studies suffered from small sample sizes, but a way of really making sense of them is to conduct a meta-analysis where we can combine the data across multiple studies. When we did this, and so did another group, we did in fact found that cannabis-using patients with schizophrenia do show better cognitive performance than patients who don't use cannabis. Now that is really counterintuitive, doesn't match what we're finding in terms of brain structure, and it's not at all what we would expect. If healthy individuals who use cannabis heavily show poorer cognitive performance, and people with schizophrenia are already compromised in cognitive function, one might for sure expect that cannabis would make things worse, and that's not what we're finding. So we've all of us in this field have been struggling a little to explain this, and the best explanation we've been able to come up with really is that this association between better cognitive performance and cannabis use in schizophrenia patients might be driven by a subgroup of neurocognitively less impaired, that is, less vulnerable individuals who only develop psychosis after a relatively early initiation into cannabis use. That is, people who may not otherwise have been as likely to develop a psychosis and only did so because they used cannabis and because they did so at a fairly young age. So they are less vulnerable to the development of schizophrenia per se, and therefore better functioning perhaps than those patients who developed schizophrenia without having the trigger of cannabis. However, we also found that most of the studies included in their samples of cannabis-using patients, those who had only used cannabis for perhaps only a few times. And if we looked at some of the studies that did indeed show poorer performance in cannabis-using patients, we found that that was associated with recent use. Those who were still using cannabis showed poorer performance. It's really a very complex area, and I think we need a lot more research. We certainly should not be advising patients with schizophrenia that they should use cannabis and that this might be good for them. That is very much the wrong message to give, and we know that cannabis exacerbates the symptoms of psychosis, results to greater relapse of psychotic episodes, and poorer outcomes in the long term. So there's a lot more work to be done to really understand what's going on here. Okay, so coming back now to my subheading of the good, the bad, and the unknown. So just about everything that I've presented so far is pretty bad for cannabis. It's not a very good story at all. In terms of the unknown, well, there are many questions always unanswered, many more questions that arise from each study that we do, much more research that we need to conduct in order to understand fully the effects of cannabis on different individuals and at different time points in their life. But then, is there anything that is actually good? Well, cannabis plant matter is comprised of a number of different compounds. THC is the primary psychoactive constituent. It is the one responsible for making us feel intoxicated. It is most associated with most of the adverse effects that I've talked about on brain structure, function, cognition, symptoms. So THC is thought to be the bad guy. But there's another compound in cannabis plant matter that is increasingly becoming very interesting, and that is cannabidiol, or CBD. It's abbreviated as CBD, and this is thought to perhaps be the good guy. So THC is anxiogenic, paranoia-inducing, psychotogenic, cognitively impairing. When THC is administered to healthy individuals and people with schizophrenia, we see transient exacerbation of core psychotic symptoms and cognitive deficits. Cannabidiol, on the other hand, has been demonstrated to have anxiolytic and, in fact, antipsychotic properties. So, in particular, there was a study published in 2012 in Translational Psychiatry by Markus Levicki's group in Germany, administering cannabidiol to schizophrenia patients and comparing it to another commonly prescribed antipsychotic. And they found that cannabidiol showed similar efficacy in terms of reducing psychotic symptoms and had fewer side effects. So this is particularly interesting. In other studies in healthy individuals, the acute administration of THC versus cannabidiol has shown opposite effects on specific brain functions and symptoms. And studies of proportional exposure to each compound suggest an amelioration of the adverse effects of THC by CBD, and that is psychotic, cognitive, and appetitive effects. How do we look at proportional exposure to each compound? Well, you can do that by analyzing hair samples, which will give you the quantification of THC and CBD from exposure at least over the last 3 months, depending on how much hair you can analyze. Now, when we did that and looked at the presence of CBD in hair samples, we indeed found that one of our studies, the group that had been exposed to CBD, so of course they were also exposed to THC because they smoked cannabis, but when they were exposed to CBD, they showed significantly larger hippocampal volumes than the group that was exposed only to THC. And in fact, the group with THC and CBD did not differ from controls. So this does suggest some protective effects of CBD. Now, why is this important? Because the cannabis that we see on the streets today has been selectively bred to be high in potency with THC, and that has caused CBD to be selectively bred out of cannabis plant matter. So in fact, there's very little CBD in cannabis plant matter at all today. And given these studies that have been coming out in recent years, there are some campaigns to reinstate CBD back into cannabis to protect against some of these adverse effects of THC. So clearly we need a lot more research to really understand what these compounds are doing. And that has led to a recent randomized controlled trial of administration of these cannabis compounds, THC and CBD, as well as both compounds in combination, to healthy humans, where we've looked at their effects on symptoms, cognition, brain function, including brain electrophysiology and neuroimaging. So in this randomized controlled trial, we've administered 5 different drug conditions, where we administer the drugs through a volcano vaporizer, so people inhale the vapors of the pure compounds. And the 5 drug conditions have been placebo, THC on its own, high-dose CBD on its own, and then THC combined with CBD at 2 different levels of CBD. The reason for this is it's these particularly high doses of CBD that have been shown to have antipsychotic and anxiolytic properties when administered to humans in oral tablet form. However, you would never see such high doses of CBD in cannabis plant matter, even in the good old days when cannabis used to contain CBD. It was more likely to be in the proportions of about 2 to 1. So that's why we've administered this low dose of CBD in one of our conditions, and in another condition we've combined high-dose CBD with THC to examine whether it may indeed ameliorate. And we've looked at a range of EEG markers and neuropsychological cognitive markers. We are looking at heart rate and blood pressure, taking multiple blood samples over the course of several hours. We administer the main dose of the drug compounds at this time point here, which I hope you can see, and we give several top-ups over the course of the experiment and measure a range at different time points. We measure intoxication, visual analog scales for a range of subjective effects, psychotic symptomatology and mood, and we'll also be looking at the effects of genotype in moderating the effects of these compounds on different individuals. We have 2 different groups that we're examining, regular cannabis users and non-naive, non-user controls, or light infrequent cannabis users. We've just completed this entire study now and are moving on to our next phase of this study, which is to look at neuroimaging, where we'll be administering these compounds within a scanner and looking at brain chemicals and connectivity throughout the brain. So what I'd have to say, we've started to just prepare preliminary data for presentation at conferences from the outcomes of this study. And I'd have to say that it's pretty early days in terms of thinking that CBD is the wonder drug. We're finding quite different effects in regular users and non-regular users. In terms of low-dose CBD, we are actually finding a potentiation of some of the adverse effects of THC, and that matches an animal study that came out of Ian McGregor's lab, where they also found potentiation by low doses of CBD. So I'm sure, as many of you know, these compounds and cannabis in itself is very complex, and I really think we need to understand these interactions a lot better before we can start telling people that CBD is good for you. So just to sum up and conclude then, the main take-home messages from this presentation are that long-term, very heavy cannabis use leads to cognitive deficits, brain structural changes, and subclinical psychotic symptoms that resemble schizophrenia. Because we've seen this in so many studies and in people who are very carefully screened for psychotic disorders, we suggest that everyone is in fact vulnerable to these kinds of adverse effects if cannabis is used heavily enough for many years. So even if people may not develop overt psychosis, they may develop schizophrenia-like conditions in the brain. We think that the adolescent brain is more vulnerable because it is a critical period of neurodevelopment. In terms of recovery of cognitive impairment, brain structure, brain function, as I said, this is still very uncertain. We do have some preliminary data at the moment that does suggest that the hippocampal volume reduction we observed may recover sometime between maybe 6 and 18 months after cessation of use. But I also heard a presentation in New York just last week from another group who were looking at adolescents in particular and found that the reductions in volume in other brain regions that they were studying did not recover after about 18 months of abstinence. So still a lot of work to be done there in terms of recovery of function. And we do, of course, think that there are individual differences in response to cannabis, and we need to understand these better. So clearly some people are much more impaired than others. Some people may be more amenable to recovery than others. It may depend on genotype. It may depend on a range of factors which we need to understand better. And it'll be multidisciplinary efforts from a range of different kinds of teams employing a range of different techniques to really further our understanding of vulnerability factors and also mechanisms by which cannabis might trigger psychosis or lead to some of these other adverse cognitive effects. So I think that we always need to consider age when we examine the long-term effects of cannabis. And hopefully most of you can read that cartoon which says, as Alf grew older, he began to have trouble with his joints. So I'll leave it there and just acknowledge many, many people that I've worked with over the years and the research that I've presented today, and also the funding agencies that supported our research. So thank you very much for your attention.